Assalamualaikum and good evening. My name is Syed Naufal bin Safami and my metric number is 198390. I'm from Group 8. Today, I will explain what I understand from cDNA library construction and screening part 5. So, from what I understand from this slide and this picture is that we want to, before transfection can happen, it means that we want to introduce foreign DNA into the bacteria or into the cell by using virus or phage. Okay, in this case, we use bacteriophage. So as you can see that, let me zoom in first. Okay, as you can see that we have complementary DNA, where our DNA on our gene of interest that are cut by such enzyme, and then we have bacteriophage. And bacteriophage, we extract the DNA and we cleave so that we can ligate the complementary DNA into the lambda plasmid, lambda DNA. Okay, then after the ligate, it will produce recombinant DNA. After we produce recombinant DNA, we package it back into the phage. Then it called hybrid phage. Okay, as you can see here. So after that, transfection can occur. As you can see in this picture, one virus infect the bacterium, and then it inject the recombinant DNA into the lysis, into the I'm sorry, into the bacteria, and then the bacteria will package it and produce all the components for the phage, and after that occur. Lysis happen. As you can see, it bursts out and release uh, release the phage that are produced. So as you can see, if we look it in our using our naked eye, we can see the plaque with forming in the lawn of bacterium. So as you can see this lawn of bacterium, which are full of bacterium, there will white spot or almost like colorless spot will produce on the lawn of bacterium. This indicate that the bacteria on the spot is already died because the lysis happened. So from what scientists can benefit from this is that it can use to amplify the library, and it can after that we can use this 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 plague to further or to further our research or to continue for another experiment that are required for this complementary DNA. Okay, so that's what I understand. And next, my friend Ilya will explain all about why phage is better as cloning vector compared to plasmid. Okay, so that's all. Thank you.
Assalamualaikum Doctor. My name is IYLIA and my metric number is 199353. So for this part, I'll be explaining the differences between bacterioplasmid and bacteriophage lambda. So, plasmid is actually bacterial DNA that is not part of the main circular DNA chromosome of the bacterium. It is small, circular DNA molecule that usually contain genes for, let's say, antibiotic resistance. And it is quite common, it is really common for cloning vector. So I've made a simple table to differentiate between plasmid vector and lambda vector. For the origin, of course, plasmid vector is from bacteria and lambda vector is from bacteriophage. As for the genetic material, plasmid vector has circular double-stranded DNA. Meanwhile, lambda vector has linear double-stranded DNA. For the examples of plasmid vector are the most common one, PBR322 and PAC19. Meanwhile, for lambda vector is lambda phage. So, lambda vector being a bacteriophage, they contain code called capsid. Meanwhile, plasmid vector does not have any code. As for the size of insert, for plasmid vector, the insert could be up to 10 kilobases. However, for lambda vector, we can go up to 25 kilobases, which means the inserts can be larger in lambda vector compared to plasmid vector. So for the steps of penetration, plasmid vector begins with the preparation and then insertion, then transformation which is the uptake of DNA material by the bacterial cell, then screening, and lastly growing and isolation. For lambda vector, we begin by preparation and then insertion, next packaging as explained by my friend before this, packaging into the hybrid phage, and then continue with transfection of the bacteriophage, of the hybrid phage, into bacteria, into chosen bacteria, and last but not least, plating. As for the position of insert in vector, plasmid vectors, for the insert, it is inserted into the multiple cloning sites. So within the multiple cloning sites, there are a lot of restriction sites there. As for lambda vector, the coupling between lambda and the material, the inserts, happens in the center portion of the vector genome. As for the type of plate, so usually for plasmid vector, we use ampicillin agar plate or Lexi gene agar plate. As for lambda vector, we use the lawn plate. So all of this I obtained from past lecture notes and also from this journal, Bacteriophage lambda as a cloning vector, from the Microbiological Reviews journal. So that is all from my part. Thank you, Doctor.